Mutation is a permanent change in a DNA sequence. Mutation can result from DNA copying mistakes made during cell division, exposure to ionizing radiation, and exposure to chemicals called mutagens or infection by viruses.
For mutation, mutation is a permanent alteration of a nucleotide sequence of a gene. Mutation also a physical event. A single base pair change in a nucleotide sequence of a gene is called a point mutation. Sickle cell anemia, hemophilia, cystic fibrosis, Klinefelter syndrome, and Turner syndrome are results of mutations.
For polymorphism, polymorphism is a presence of more than one allele at the particular locus in a particular population. It is a population event. A single base pair change in the nucleotide sequence of a gene is called a single nucleotide polymorphism. Human gender and ABO blood group is the result of polymorphism.
The causes of gene mutation. There are two main causes for gene mutation. The first one is inherited. The mutation inherited from the parent to their children. Another one is acquired, and it divides into two factors, which are environmental factors and in-body factors. For environmental, it caused by three factors. First, physical, for example, radiation. Second, biological, for example, bacteria Helicobacter pylori. Third, chemical, such as harmful clinical reagent. For in-body factors, it is split into two, which are during DNA replication and DNA transcription.
Mutations that occur in somatic cells are known as somatic mutation. This type of mutation occur because of the alteration in DNA that occur after the conception in specific cells, except germ cells. Due to the reason where it not occur in germ cells, this mutation will not be passed along to the next generation by sexual means.
Germline mutation is an inherited mutation. This mutation exists in the germ cells, which is egg and sperm cell. Mutations in these cells are the only mutation that can be passed on to the future generation when either a mutated sperm or oocyte come together to form a zygote. Mutation is present in all the cells of his or her body.
The types of mutation include first, substitution or point mutation, which is a replacement of a single nucleotide by another. Point mutation are the most common type of mutation, and there are two types, which are transition and transversion. The second mutation is missense mutation. This type of mutation is a change in one DNA base pair that results in the substitution of one amino acid for another in the protein made by a gene.
Next is nonsense mutation. A nonsense mutation is also a change in one DNA base pair instead of substitution one amino acid for another. However, the altered DNA sequencer prematurely signals the cell to stop building a protein. And lastly is silent mutation. Some mutations that change DNA base do not have any effect on the sequence of amino acid in the protein.
Numerical abnormalities. It means the variation in chromosome number. Aneuploidy. It can divide into two, trisomy and monosomy. Polyploidy. Chromosome number that is multiple of the normal haploid chromosome sets. Triploidy. What is the example for autosomal trisomy, Down syndrome, and Bantu syndrome?
Next, we move to structural abnormality. It is due to the changes of structure within chromosome. There are many types of structural abnormalities, which are translocation, deletion, duplication, inversion, ring, and isochromosome. Now, we move to the explanation of deletion and duplication, translocation and inversion, rings and isochromosome.